Last week we extracted DNA. The week before that, sterile technique. Every Monday on this show, one technique. Start to finish, no filler. I'm Kelpie, this is Method. Monday, and I'm covering gel electrophoresis today specifically, because right now, in my shed, I have DNA extracted from 17 environmental sample sites, and I need to read what's in them. That is a longer story. It starts here. You've done the extraction, you have a tube. Congratulations. It looks exactly like water. You have no idea if it worked, how much you got, whether it's intact or degraded, or whether it's contaminated with RNA. Gel electrophoresis is how you find out, and if you skip this step, every downstream experiment you run is operating on faith.
Every DNA molecule carries a net negative charge, uniformly distributed along the phosphate backbone. In an electric field, it migrates toward the positive electrode. The Agarose matrix acts as a molecular sieve, a three-dimensional mesh of hydrogen-bonded polysaccharide chains. Small fragments thread through the pores quickly. Large fragments crawl. The result is size-based separation resolved in space across the gel.
The gel box, the power supply, the transilluminator, $400 total, roughly. That is the cheapest instrument in molecular biology that tells you whether $200 of extraction reagents actually produced something worth using.
The log-linear relationship between fragment size and migration distance is what makes the latter useful. Plot the log of each ladder band's molecular weight against its migration distance. You get a straight line. Your unknown bands fall on that curve. This is why gel electrophoresis survives in a world where sequencing costs $1 per kilobase. It tells you what's there and roughly how big it is before you spend the sequencing budget to find out what it is exactly.
It is mildly elegant that a technique for seeing DNA requires destroying a small portion of it.
A small aliquot, 2 to 5 microliters from a diluted sample. The rest stays in the tube.
Subscribe. Gel electrophoresis costs $2 per sample. Skip this episode and you'll spend $20 in failed downstream reactions figuring out what this would have told you for free.
And if this is your first Method Monday, we build methods in sequence. Last week, DNA and protein extraction. Next week, PCR. Everything we cover today is a prerequisite for that. Stay. Equipment. Agarose first. Molecular grade, low EEO. EEO is electroendosmosis. High EO, Agarose generates a counterflow that drags water backward through the gel during the run, distorting your bands. The bottle says Agarose, and it will quietly ruin your resolution. Get the molecular grade version. I learned the difference on a borrowed gel box at 2 a.m. You don't need to learn it that way.
I use the bacteriological grade. We have a lot of it.
Where did you get a lot of bacteriological Agarose?
The microbiology department was throwing it out.
It is mildly characteristic that this answer raises more questions than it resolves.
Running buffer. TA trisacetate EDTA is the default. Covers genomic DNA, PCR products, restriction fragments in the normal size range. TBE, trisborate EDTA, gives you sharper resolution for small fragments under 500 base pairs. If you're resolving microsatellites or oligos, use TBE. For everything else, TAE.
The borate in TBE inhibits some downstream enzymatic reactions. If you're excising a band and ligating it directly, use TAE. The ETA in both buffers chelates divalent catechins, which inhibits DNA's activity and prevents metal-catalyzed DNA hydrolysis during the run. That is not incidental. Long runs without EDTA in the buffer leave your DNA progressively more vulnerable to degradation.
Stain. Cyber-safe. Non-mutagenic. Reasonable sensitivity. No disposal drama. Ethidium bromide if you have hazardous waste infrastructure. More sensitive, cheaper, works better. It's a mutagen, but not a terrifying one if you handle it with gloves. Precast stain into your gel during pouring saves the post-stain incubation step. That's 30 minutes you don't wait. Ladder. Pick one that covers your expected range. Running genomic DNA, you need a ladder with bands up to 20 kilo bases. Running PCR products, a 100 base pair or 1 kilo base ladder. If your bands fall off the top or the bottom of the ladder, you're extrapolating. Extrapolating from a gel standard curve is approximately the same as guessing, but with more confidence than you deserve. Step 1. Cast the gel. The percentage determines everything about what you can resolve. 0.8% for high molecular weight genomic DNA, 10 kilobases and above. 1% for general PCR products and restriction fragments, 500 to 5,000 base pairs. 1.5% to 2% for smaller fragments under 500 base pairs. 3% only for microsatellites and very small fragments under 300 base pairs. Most people run 1% for everything. It mostly works. Mostly is where experiments go to die.
The percentage directly sets average pore diameter. At 0.8%, the mesh is loose enough for high molecular weight DNA to migrate, slowly but measurably. At 2% and above, fragments over 5 kilobases stall at the well. The pore diameter is physically smaller than the end-to-end length of the molecule. You get a bright band at the well and no migration downward, and if you don't know the cause, you will troubleshoot everything except the variable that is actually wrong.
I use 3%.
For what?
Genomic DNA, 15 to 20 kilobases.
Right, stop! Don't run that! Your molecule cannot thread through a 3% matrix! You'll get a smear at the well and nothing below it.
The end-to-end length of a relaxed 20 kilobase DNA fragment is approximately 7 micrometers. Average pore diameter in a 3% agarose gel is roughly 80 to 100 nanometers. You're attempting to move a 7 micrometer molecule through a 100 nanometer opening. The molecule cannot reptate. It stalls. More agarose, in this case, provides less information.
It is mildly counterintuitive that more of the matrix gives you less data about the molecule.
Recast the gel. Yes, that is another 30 minutes. I've run the time cost calculation on just run it and hope versus recast and wait. Hope costs more every time.
Step 2. Load. Loading dye is not optional. It does 3 things. Glycerol adds density, so your sample sinks to the bottom of the well before it can diffuse back into the buffer. Bromophenol blue tracks the slow migration front. Co-migrates with roughly 300 to 500 base pairs on a 1% gel. Silene cyanol tracks the fast front at around 4 kilobases. These 2 markers tell you exactly when to stop the run without lifting the lid.
The wells are filled with running buffer. Without loading dye, your sample density is approximately equivalent to that buffer. Pipetting into a buffer-filled well introduces your sample into a fluid medium where diffusion dominates over sedimentation. The sample spreads laterally instead of sinking into the well. Some portion enters the gel, some doesn't. Reproducibility is eliminated before the run even starts.
Done.
Did you add loading dye?
No,
All of it?
all of it.
Salvage. If you have remaining sample, add loading dye now and load into unused wells. What's already in the gel? Some of it sank by density alone, some didn't. You'll see no dye front. You won't know when to stop, and you'll have to guess a run time. Add loading dye to everything before it touches the well. 10 seconds of prep every time, no exceptions.
It is mildly instructive that the component everyone omits most often also does the most work per microliter.
Step 3. Run the gel. 80 to 120 volts constant voltage. 100 volts is the correct answer for standard work. Higher voltage looks faster and gives you worse resolution, joule. Heating diffuses your bands and can melt the gel at the edges. I have run a gel that bubbled. The bands were unusable. 100 volts. Run until the bromophenol blue front is 1 to 2 centimeters from the gel edge. Not off the edge. Once small fragments run off, they're gone.
Voltage sets the electric field across the gel. At excessive voltages, joule heating increases the thermal motion of DNA, and the agarose matrix becomes slightly more fluid. Band diffusion increases, and the log linear relationship between migration distance and fragment size, which is the basis of accurate sizing, breaks down. Stay within the range where that relationship is linear.
It's running.
Which direction are your leads?
The black one is toward the wells.
Stop the run right now!
45 seconds.
Reverse the leads. Red toward the front of the gel. DNA is negatively charged. It runs toward the positive electrode. Red is positive. Run toward red. If you run this another 4 minutes, the wells will be empty, and your DNA will be in the buffer tank. You'd be re-extracting.
The phosphate backbone carries a fixed negative charge independent of sequence, fragment size, or organism. Under no standard electrophoresis conditions does DNA migrate toward the negative electrode. If wells are clearing without corresponding band formation in the gel, the first check is always polarity. It is the most common cause of complete run failure.
45 seconds of reversed polarity, and you recover the run. 45 minutes, and you re-extract. I've priced both outcomes. One is significantly worse.
Step 4. Stain an image. If you precast the stain into the agarose, which I do, skip the incubation and go straight to the UV transilluminator. If you're post-staining, incubate the gel in fiber-safe or ethidium bromide solution for 20 to 30 minutes, then rinse in water for 15 minutes to reduce background. Image on UV. Bands light up fast. Photograph before they diffuse if you're using ethidium bromide, which diffuses out of the gel over time.
Both intercalating stains function by inserting between stacked base pairs of double-stranded DNA. The bound dye fluoresces under UV far more intensely than the free form. Fluorescence intensity is proportional to the mass of DNA in each band. A brighter band contains more DNA. The relationship is not precise enough for accurate quantification, but it is informative. You can immediately see relative yields across lanes and identify lanes with little or no DNA.
Good gel result for genomic DNA. One tight band sitting high on the gel, 15 to 20 kilobase range. Clean lane below it. No diffuse smear running toward the bottom, which means no degradation. No low molecular weight smear near the 100 base pair region, which means no RNA contamination. If you ran RNase A during extraction, which we covered last week, the lane should be clean. If you didn't, you'll see a diffuse smear in the low MW region. That's not fatal for most applications, but know what it is.
A smear running from the main band downward is diagnostic for fragmentation. The original high molecular weight molecule has been cleaved into fragments of varying sizes. They separate during migration, producing a continuous smear rather than a discrete band. The most common causes: nuclease contamination introduced during extraction, heat exposure during lysis, or repeated freeze-thaw cycles of the stored extract. A partial smear, only below the main band, not reaching the bottom, indicates partial degradation. The sample was intact at extraction and damaged in storage.
Wait, if genomic DNA barely moves, how do you know the run worked at all? What if the gel just didn't do anything?
The ladder. If the 20 kilobase ladder band migrated 1 centimeter into the gel and your sample band is sitting in the same position, that's valid migration for that molecular weight. The ladder is your run control. It tells you the gel ran correctly under the same conditions your sample experienced.
If the ladder bands resolve correctly and your sample lane is completely empty, you either didn't load or loaded without glycerol and your sample is in the buffer. If the ladder didn't resolve, the gel failed and no other interpretation is valid. Run your ladder in every gel every time without exception.
It is mildly clarifying that a smear tells you more about what went wrong than a clean band tells you about what went right.
Here is where gel electrophoresis stops being quality control and becomes analysis. Digest your DNA with restriction enzymes. They cut at specific recognition sequences, generating fragments of defined sizes. Run those fragments on a gel, and the band pattern is a fingerprint. Different organisms, different samples, different individuals, different patterns. That's RFLP, restriction fragment length polymorphism, genetic fingerprinting before sequencing existed.
A restriction enzyme recognizes a specific 4 to 8 base sequence and cleaves both strands at defined positions. Agarose recognizes 5' GATC 3' and makes a staggered cut, generating 4 nucleotide 5' overhangs, sticky ends that enable directional ligation. Digest 2 samples with the same enzyme. If they're identical in sequence at the restriction sites, the band patterns are identical. Different organism, different genomic architecture, different pattern. The gel doesn't read the sequence, it reads the map.
RFLP predates affordable sequencing by 2 decades. Before a genome cost $1 per kilobase, this was forensics, pathogen identification, and GMO verification. Some diagnostic labs are still running RFLP panels because the gel infrastructure is paid for and the assay costs less than sequencing per sample at scale.
This dual function is what makes gel electrophoresis irreplaceable, even as sequencing costs drop. Every gel you run to check an extraction can also serve as the analysis if a restriction digest step is added upstream. It is not just verification, it is the foundation of fragment-based molecular analysis.
Pro tip the manual skips. Track your Agarose batch number. Migration rate shifts between lots. Same brand, different bag, subtle change in polymer chemistry. Your bands move a few hundred base pairs. I spent 2 days troubleshooting a migration artifact that was a new batch of Agarose. Write the lot number in the gel image file name. In 6 months, you'll open a folder of gel photos and be able to tell which ones are directly comparable.
The EEO value varies between lots as well. Subtle changes in the polymer affect how water migrates through the matrix under an electric field, and this contributes directly to the migration rate shifts Kelpie is describing. It is not just empirical variation, there is a physical mechanism, and tracking the lot number is the simplest way to account for it.
Right? The experiment. 14 months ago, I started collecting environmental samples. Soil and water, 17 sites along an industrial drainage corridor. Everything extracted by the protocol we ran last week. Separate DNA and protein fractions labeled, stored at -80. 6 weeks ago, I started running RFLP panels on the DNA fractions. EcoRI and HIN3 double digest. Together they give a diagnostic fingerprint for whatever organisms are living in those samples. The Site 7 samples, drainage corridor, eastern sector, have bands that shouldn't be there. Consistent bands, same molecular weight, same pattern, across 6 independent collections spanning 8 months. Not contamination, not a degradation artifact. A repeating pattern I can't match to any published restriction map.
I ran the replicates last night. 3 independent digests from the Site 7 batch C samples. The anomalous band is at approximately 2.3 kilobases on every gel. I have the photographs. I also ran batches D and E while I was there.
It is mildly consequential that the unauthorized experiment now has a reproducible result.
Closing tip. Tay loses buffering capacity during long electrophoresis runs. pH shifts as the buffer exhausts, migration rate becomes non-linear, and your size estimates drift off the standard curve without any obvious sign that something is wrong. If your run goes longer than 90 minutes at 100 volts, make fresh buffer. 90 seconds to dilute from stock. That habit alone will prevent 3 confusing gel results a year. Fresh buffer every run. That's gel electrophoresis. You pour the agarose, run the current, and read every band. The ones you planned for and the ones you didn't. The unexpected bands are the ones that mean something. Next Monday, SDS page. Polyacrylamide gels. Denaturing conditions. And we're running the protein fractions from those same Site 7 samples. The ones we extracted alongside the DNA last week. If the DNA restriction pattern is anomalous, I want to know what the protein profile looks like too. To call yourself a mad scientist, you need to read every layer of the experiment. Method Monday. Every Monday.